Hello book lovers, this is Vanapalli Harshiki, you are seeing Family Notes. Today our topic is Intrinsic Clotting Cascade and Extrinsic Clotting Cascade. Here we can see both intrinsic pathway and extrinsic pathway and how they are interconnected. Let us see about Intrinsic Clotting Cascade. The intrinsic pathway is also called as contact activation pathway. That means it will be activated when any bacterial infiltration or any abnormal physiology such as hyperlipidemic states or any other physiological conditions that can trigger the activation of intrinsic pathway. The intrinsic pathway requires the clotting factors 8, 9, 10, 11 and 12. This also requires the proteins like prekallikrein and high molecular weight kininogen as well as calcium ions and phospholipids secreted from platelets. Each of these pathway constituents leads to the conversion of factor 10 into factor 10A, that is inactive factor 10 to active factor 10. The initiation of the intrinsic pathway occurs when prekallikrein, high molecular weight kininogen, factor 9 and factor 12 are exposed to negatively charged surface of the cell. This is termed the contact phase and can occur as a result of interaction with the phospholipids. This contact activation of the intrinsic pathway can also occur on the surface of bacteria and through the interaction with urate crystals, fatty acids, protoporphyrin, amyloid beta and homocysteine. The assemblage of contact phase components results in the conversion of prekallikrein to kallikrein, which in turn activates factor 12 to factor 12A. Now this factor 12A then activates factor 11 to factor 11A. Factor 12A will also hydrolyze more prekallikrein to kallikrein, establishing a reciprocal activation cascade. Kallikrein acts upon high molecular weight kininogen, leading to the release of bradykinin, a potent vasodilator. In the presence of calcium ions, the factor 11A activates the factor 9 to factor 9A. The factor 9 is a proenzyme that contains vitamin K dependent gamma carboxyglutamate residues, whose serine protease activity is activated following calcium ions binding to these GLA residues. Active factor 9A cleaves the factor 10 to activate it to the factor 10A. The activation of factor 10A requires the assemblage of the tenase complex, that is calcium ions, factor 8A, 9A and 10 present on the surface of activated platelets. One of the responses of platelets to activation is the presentation of phosphatidylserine and phosphatidyl inositol on their surfaces. The exposure of these phospholipids allows the tenase complex to form. The role of factor 8 in this process is to act as a receptor in the form of factor 8A for factors 9A and 10. Factor 8A is termed as a cofactor in the clotting cascade. The activation of factor 8 to factor 8A occurs in the presence of minute quantities of thrombin. As the concentration of thrombin increases, factor 8A is ultimately cleaved by thrombin and inactivated. This dual action of thrombin upon factor 8 acts to limit the extent of tenase complex formation and thus the extent of the coagulation cascade. Now let us know about extrinsic clotting cascade. Activated factor 10A is the site at which the intrinsic and extrinsic coagulation cascades converge. The extrinsic pathway is initiated at the site of injury in response to the release of tissue factor, that is factor 3. and thus it is also known as the tissue factor pathway. That means extrinsic clotting cascade is also known as tissue factor pathway. The tissue factor is a cofactor in the factor 7A catalyzed activation of factor 10. Factor 7A, a GLA residue containing serine protease, cleaves factor 10 to factor 10A in a manner identical to that of factor 9A of the intrinsic pathway. The activation of 7 factor occurs through the activation of thrombin or factor 10A. That means the ability of the factor 10A to activate factor 7 creates a link between intrinsic and extrinsic pathways. An additional link between the two pathways exists through the ability of tissue factor and factor 7A to activate the factor 9. The formation of complex between factor 7A and tissue factor is believed to be a principal step in the overall clotting cascade. If you like the video, please subscribe our channel.